Hello, my name is Carl, and in this video I'm going to briefly discuss point mutations and their effects on amino acid sequences. The mutations I'm going to cover in this video are called SNPs, or single nucleotide polymorphisms, which is a way of saying that these mutations only involve a change in one single base pair of the genetic sequence. This is our DNA sequence of interest that we're going to be looking at. For this problem, we are starting with the knowledge that we are looking for a 5 amino acid long exon. To figure out exactly which part of the sequence this is, we're going to start with looking for our ATG start codons to set our reading frame and figure out which parts will actually be transcribed into an amino sequence. It appears we have 2 start codons, so we're going to count the number of codons after till we get to a stop codon and see which one has 5 amino acids. It looks like our bottom strand is actually going to be our coding strand and our top strand is going to be the template strand. I will go ahead and transcribe this sequence into RNA and affix the appropriate amino acid sequence. For codons that can have a third position wobble, they have been marked appropriately. I will put back up the original DNA sequence and compare it with our first mutated sequence. This involves a transition of a A to a G and results in arginine becoming glycine in the amino sequence. This single change in an amino acid within the sequence is called a missense. Here I have reversed the previous mutation and added in a new mutation, which is a transition from a C to a T. Because of the third codon position wobble, this actually results in no change to the amino sequence. When this happens, it is called a silent mutation. As before, I have undone the previous mutation and added in a new one, this time a transversion from an A to a T, which results in a premature stop codon. This premature stopping is called a nonsense mutation and can often result in a truncated protein. The previous mutation is undone once again, and we see our last mutation. Here we see a transversion from an A to a C, which results in the stop codon changing to serine. When a mutation eliminates the stop codon, this is called a read-through mutation and can result in the protein being changed as the genetic sequence continues to be translated. So what actually happens when mutations change this amino acid sequence? Well, we can then sometimes have changes in the phenotype, but oftentimes we don't, and this is what's called a neutral mutation, where whatever change was made doesn't end up changing the phenotype. But sometimes we do have a change, and we can have a loss of function mutation. A loss of function mutation can mean that some protein or pathway has lost the ability to do what it did before, and then can end up in maybe genetic disease or even a loss of fitness. Sometimes this is good though, and we get a gain of function mutation, where the change that was made influences the phenotype in a way that actually adds to the fitness of the individual. So that's it. Thank you for watching, and hopefully now you understand a little bit more about point mutations.